Thanks so much for having me. It's really lovely to meet you all and to be here today. I think Julian he gave me a topic basically that he thought, because I've got obviously like a very wide range of interests, mostly to do with genomics. But one thing I have worked on quite a lot in the time since I finished my PhD is things to do with return of results in genomics, and particularly things like incidental findings and secondary findings. And I think he thought there would be some overlap with some of the work that was being done here. So hopefully you find it interesting. You said like I have an affiliation with KU Leuven. Actually, when I finished my PhD, I moved to Leuven in Belgium for my very first postdoc. And that was what got me onto the path of looking at return of results in genomics. So I feel like I need to acknowledge that because that was sort of the basis of all my interest in the area. So I'm going to present to you today quite a lot of different studies that I've done that all sort of touch on this topic. So it might, hopefully there's a stream that runs through the whole thing about incidental findings. But it's a whole bunch of different studies using different methods that I've put together over the last 7 years since I finished my PhD. So hopefully it's interesting. But I wasn't sure what kind of background people had, and I thought maybe not everyone is familiar with genomic technologies. So I just thought I'd give you a little bit of a rundown of what I'm talking about when I'm thinking about genomics. So genomic sequencing refers to a range of different next generation sequencing technologies. So we have whole genome sequencing, which is where you have basically as much of the genome as we can sequenced. And then you can have exome sequencing, which is where we just sequence the bits that code for different proteins. So it ends up being roughly about 1 to 2% of the genome that is sequenced using exome sequencing. And then we have gene panels. And they can be either sort of set panels where you actually only look at those particular genes using the technology, or it can be virtual panels where you actually sequence the whole exome, but you just do a filtering process so that you only look at the genes that are of interest. And that is usually filtered by the condition that that person is presenting with, so what the clinical picture looks like. So when we're talking about gene panels, that's what we're referring to. So initially, genome sequencing really just started in the research setting. So about 2006, this new technology of massive parallel sequencing was developed, and they realised that this was really pivotal in terms of its ability to change the way we look at genomes and help identify disease. So it was initially was sort of looking at rare diseases and cancers, but now it's really starting to be used in a whole bunch of different other areas and specialties within medicine. So cardiology, nephrology, neurology, etc. And that has huge implications for clinical care because people who are not trained in genetics are starting to be issuing these tests and referring people for these tests and doing the consent process for these tests. And so that means that we need to be thinking about how this mainstreaming is impacting clinical care. So there are lots of different contexts that genomic sequencing can be used for. So obviously there's the diagnostic setting where you might have children who have a rare disease or in the acute care setting, so critically ill children. Now we have rapid genomic sequencing where kids who are in the NICU and PICU can be sequenced within a few days to try and identify the cause of their genetic condition, and that's really helping influence treatment. But it can also be in adults, often in things like cancer or renal conditions or heart conditions. But it can also be used in the screening context. So we've got adulthood, like expanded carrier screening now, where you can try and figure out if couples both carry a recessive allele. That means that they're at risk of having a child that's affected with the condition. We've got newborn screening, where genomics is starting to be used, mostly in the research setting, but it's becoming more and more prevalent, particularly in the US. And then in prenatal setting as well, they're starting to use exome sequencing more and more in terms of identifying genetic conditions in babies that present with fetal abnormalities on things like ultrasounds. So obviously there are huge benefits from this technology. So it does increase the rate of gene identification. It decreases the time to a diagnosis, and then that means that there's lots of other tests that aren't necessary as well. So for example, if you do exome sequencing or genome sequencing as a first-line test in a critically ill kid, then it means we probably don't have to do things like invasive tests like muscle biopsies and stuff, which is obviously expensive but also really painful for kids. And it's also becoming more cost-effective. So there's more and more evidence that comes out that if you do this testing early, then it means that you actually save costs in the long run because you're doing less other tests as well, and because it changes healthcare management. But it does create some ethical challenges, and that's why I'm going to sort of focus on some of today. So one of the things that is ethically challenging is that genomic sequencing can actually increase the uncertainty compared to doing other types of testing, and I'll explain why. So there are lots of different types of findings from genomic sequencing. So one is that you can get no result, or you might get a benign variant, which means that it doesn't actually cause the genetic condition, but you see a change in the patient. And so in terms of the criteria that we use for reporting these types of findings, we refer to them as type 1 or type 2 variants, known as benign or likely benign. Then you might find the cause of the genetic condition, and so that would be a variant that is either likely pathogenic or pathogenic, and we refer to them as 4 or 5 in this class. But the trickiest thing is the variants of uncertain significance, and they're what we call class 3 variants, and they're tricky because it means that we don't have enough evidence at this point in time to be able to classify them as either benign or pathogenic. So they're kind of like in limbo land, these variants, and they might change over time because more information might be identified, but at the moment we don't really know what to do with them. And that's tricky because we don't really know whether to report them or not. And then off to the side, you've got what are either called incidental or unsolicited findings. I'm going to call them incidental findings just because I think that's the technology, the term that's most in use at the moment. And that's where you have a variant that is either likely pathogenic or pathogenic, but it's in a gene that is not related to the cause, not related to the condition that you were doing the sequencing for. So you might have an eye condition that you're doing the sequencing for, but you find you've got a breast cancer gene or something like that. So that's an incidental finding. A secondary finding is where you find something like that, but it's because you're actively looking for it. So rather than finding it incidentally or accidentally, it means you're actively looking for a list of genes that are pathogenic, that cause like certain conditions, and they're mostly cancers or heart conditions. So that's the difference between an incidental and a secondary finding. So I just said to you that it can be challenging to determine what to report when it comes to variants of uncertain significance, and I'll explain some work I've done that shows that this is a bit of a mess. We need to think about whether we report any or none. Do we only report the variants of uncertain significance that are in genes that have been already associated with that genetic condition that you're doing the test for? Or do you do it in candidate genes? So that means genes where it might look like another gene that is already been linked to that condition. So the particular nature of that gene means that it's a very feasible candidate, but there's no evidence that links it to that condition yet. The same goes for unsolicited or incidental findings. So should we report any of them or none of them? If we're going to report some of them, which ones, and how do we decide which ones to report? So I've just got a couple of scenarios to illustrate the kinds of things I'm talking about. So say we've got Peter, who's 28 years old, 28 years old. I just aged him by 20 years, sorry. Poor Peter. He doesn't have an aging condition. He's got focal segmental glomerulosclerosis, which is a kidney condition. So naturally, we're going to be looking for a change in his DNA that would cause that. But accidentally, we might identify that he's got a BRCA2 mutation, which would make him predisposed to breast cancer or prostate cancer. And so that is an actionable finding in the sense that when we know that he's got that BRCA2 mutation, we can give him extra screening to mean that he can do something about it and pick it up earlier. On the other hand, if he was found to have an ApoE4 homozygous mutation, which means that he's therefore got an increased risk of Alzheimer's disease, that is much less actionable in that there's nothing really we can do about that to prevent him getting it, apart from making him potentially exercise a bit more, but it's a lot less solid in terms of the actionability. And then we've got a childhood example. So we've got Sarah, who's 5, not 15 or 25. And so she has retinitis pigmentosa. So again, we're going to be looking specifically for genes that are related to that condition. But what if, for example, we found out that she had familial adenomatous polyposis, which means that she's got an increased risk of developing colon cancer? So that would be an actionable finding in the sense that we could, from that information, we could give her extra screening, meaning that she could potentially find out much earlier if she had any polyps that were becoming cancerous, and therefore that could be life-saving. But on the other hand, this is an adult onset actionable condition, so BRCA1 mutation. So if we found that, she would be at risk of developing breast cancer, but that's not something that she has to worry about in childhood. So we'd report that, because it means that we don't allow her the chance to decide whether or not she wants to receive that information when she's an adult. And that's some of the questions that we face when we're looking at this topic. Another ethical challenge is difficulties obtaining informed consent. That sort of goes overarching on all of these topics. So we know that informed choices require sufficient understanding, and we also know that genomics is a complex field. And so some authors, and I think one of them might be in this room, have questioned whether patients can truly give informed consent about receiving unsolicited findings or not. So just to talk a little bit about the guidelines that we have on unsolicited findings, there have been quite a few position statements around the world that have talked about whether we should be reporting these back or not. And the overarching message is that they're not consistent. So the European Society of Human Genetics in 2013, and some of these are really old now, just to flag, because I think they're actually developing a new policy statement at the moment, but I don't think it's going to be much different, to be honest. And generally the ESHG have proposed that we use a targeted approach to try and avoid finding unsolicited findings or findings that can't be interpreted. But then if you do detect them and it's indicative of a serious health condition, there is a sort of a suggestion that you should actually report that to adults, but they don't really weigh in on children so much. From another European guideline, the Eurogen Test Association, which is actually now a subset within ESHG, they again have sort of proposed more of a focused, targeted approach to try and avoid finding unsolicited findings. But basically they say that labs need to have a defined protocol about what they're going to do. So they're sort of fence sitting on that one a bit. The Canadian College of Medical Geneticists, they again sort of try and propose that you should try and filter out as much as possible to try and avoid finding unsolicited or incidental findings. But they acknowledge that sometimes you might want to do a broader analysis. So say, for example, you do a very set virtual panel to begin with, and you say, well, we didn't find the gene we were looking for. Maybe we want to expand it out a little bit more to be a bit more broad. And they say for adults, they should have a choice about whether they want to receive unsolicited findings or not. And then if we turn to actively searching for secondary findings, so that's where I said that you might have a list of, currently the suggestion is 73 genes that the American College of Medical Genetics and Genomics propose. And as I said, they're all sort of cancer and cardiac conditions. So in 2013, the ACMG made a very controversial at the time statement, and still kind of remains a bit controversial depending on where you are in the world, that we should be actively seeking to look for these genes in all patients, be they adults or children, and that they should be reported. And initially they said you shouldn't get to choose to opt out of them, but then they kind of backflipped on that a year later. But they keep updating their list every couple of years, and they have quite a lot of genes that they're adding every time. I think the French group is the only place in the world that actually has a similar kind of mentality around searching for secondary findings. Pretty much everywhere else says we shouldn't be doing it yet. And one of those groups is the Canadian College of Medical Geneticists, where they basically say they don't endorse searching for secondary findings. And then just this year, the European Society of Human Genetics came out with a very thoughtful and detailed statement where they said that they still think it's too early to be actively searching, and they call them opportunistic screening for these findings. But they basically said it's too early to do it yet because we don't have the evidence base to say that these mutations that we're finding in these patients are actually going to cause these diseases. And that's because these mutations are all in a like a selected population of people who already have that condition. So that if we're saying, well, we know that these variants cause cancer in cancer patients, but do they cause cancer in people who will maybe develop cancer in the future? And we don't really know that yet, so we need some more longitudinal studies. So they're basically saying we should be cautious until now. So I'm going to stop talking about the guidelines because they're not that exciting, and hopefully talk to you about something that's a bit more exciting, which is my own work. I find it exciting anyway. And I want to tell you about what's been happening in practice over the last sort of 7 years. So when I started my postdoc in Leuven, we were really wondering what was going on. So how were these guidelines being used in practice? And what did that mean for patients? So I wanted to do a few different studies that sort of targeted different stakeholders to try and figure out and get a picture of what was happening. So firstly, I wanted to talk to laboratory scientists. So I used purposive sampling to identify lab scientists who were using genomic sequencing. And at that stage, it was predominantly exome sequencing because genome sequencing wasn't really feasible at that stage, because this was sort of around 2015, 2016. And I used semi-structured interviews, and I talked to them about a bunch of different stuff. So I asked them about what kind of technologies they use, what kind of filtering strategies they use, and how they report and what they report, et cetera, et cetera. And I used inductive content analysis to analyse the data. So I interviewed 19 lab staff in 18 interviews. And I don't know how many people are qualitative researchers in the room, but doesn't sound like many, but it's a decent amount. And so I was really trying to look at practices in Europe, Canada and Australia, because I felt like there was a lot of research being done in the US and not much being done anywhere else. And so the lab scientists I spoke to said that some of them were only reporting variants that were relevant to the clinical question, so only pathogenic and likely pathogenic variants that related to that condition that they were asking the clinical question for. But some said they would report unsolicited findings as well. But there was a lot of variation within that. And so some said that they reported on clinical actionability. So that means can a treatment or a prevention or a surveillance be identified that can help that patient. But others had a bit more of a loose kind of an idea about what that meant. And they talked more about medical relevance. So it meant that they were thinking about what might be possible right now, but also what might be possible in the future. So could there be other things? So it was sort of more about health-related findings rather than just things that were medically actionable. Some of them said that they made decisions about how to report these unsolicited findings by discussing it with their referring clinician to see if they thought it was relevant or not to that particular patient. But most of the time there were team meetings, which we call MDTs, multidisciplinary team meetings, where they'd have committees of multiple different stakeholders, so laboratory scientists, clinicians, sometimes ethicists, lawyers, et cetera, that would help sort of each put in their expertise to try and make decisions about what was worth reporting or not. And these were often made on a case-by-case basis. Some people said that they draw on the guidelines to help them make decisions, but when I tried to pin them down and say which guidelines do you use, they didn't really have an answer, so I wasn't convinced by that. And some said that if they know the preferences of the family, that will help determine what they do. But I'm not really sure how many people actually asked what the preferences of the family were. And certainly from the guidelines, it seemed like not many organisations were trying to suggest that patients should have a choice anyway. So I felt that a more systematic approach was needed to try and do this. That was a great exploratory study and it helped me really understand what was going on. But I felt like we needed to do something a bit broader to get more of a sense of the impact that this was going to have at a clinical level. So quite a few years later, so this was in 2018, I did this study, I hooked up with a guy in Utrecht who was a laboratory scientist, and we, with the help of one of his bioinformaticians, we developed a virtual patient-parent trio. And what we did was we used the genome in a bottle background sequence, and we actually spiked it with a bunch of different mutations to try and make what would have been a very sick person in the end. But we put in, I'll show you the list of variants in a second, but we put in 8 different variants into this. And the reason for doing that was because they each had a different purpose, because we wanted to see what they would report and what they wouldn't report. So we gave them a clinical history, which was basically a child who had a very complex picture with dysmorphic features, motor delay, cognitive developmental delay, and also a mild cardiac hypertrophy. So this is the list of what we put into the genome. So you can see here that there was these two up here, I'll just point, Cornelia de Lange syndrome and spinal muscular atrophy with lower limb predominance. Actually, both together would account for that phenotype, but on their own wouldn't. So it was like a very complex picture, but we wanted to see which of those, or if they reported one or just 2 or both. We also put in a couple of different hypertrophic cardiomyopathy genes, and they're both heterozygous, so one from the mother and one from the father, and we wanted to see what they would report. And the reason for that is that one of these is on the list that would be actively searched for by the ACMG recommendations, and the other one wasn't. FLCN was a de novo mutation, so it wasn't passed on by the parents. But this is like something that can cause some renal issues. And then these two can cause familial adenomatopolyposis. But in terms of the guidelines of reporting for the ACMG, it shouldn't be reported because it's recessive, but only one of them is a pathogenic mutation and the other one is a variant of uncertain significance. And the ACMG says you should only report when you've got 2 likely pathogenic or pathogenic variants. And then this was a carrier status as well. So that's the mix of what we put in, and you can understand that when we got some reports back, I got some nasty words from some of the laboratories who said, why did you put in so many variants? And I had to explain it. And these are the 4 that are related to the unsolicited findings, so I'm going to talk about these today. But just to give you a bit more detail about what we did, I recruited through a few different channels. So I searched through OrphaNet, which is a database that they're all databases, to be honest, but OrphaNet and the Genetic Testing Registry, I went through to see who was doing genomic sequencing. And then GeneMatcher is a database that you can try and get matches with other researchers that are finding things. And so my collaborator had some connections through that as well. So we sent out a bunch of different invitations to do this. We had 2 phases to the study. The first one was that we sent these materials to the laboratory, so the clinical history that I just showed you. We put together a demographics questionnaire which really asked things about what kind of technologies they use in the laboratory, quality control mechanisms and that kind of stuff. And then we linked them to the BAM and VCF files, which is how they could download the data, the virtual patient data, for the parents and also the child in order to analyse it for the study. And we said, analyse this the way you would do a normal patient, and send us a report as though we were the referring clinician. So there was no deception or anything. They knew what they were getting into. And then we also asked them to complete the first questionnaire. So that was phase 1. And then phase 2 is when they sent all that back and we got the report. We actually sent them another questionnaire, which was asking them why they had or had not reported each different variant. And this was also a bit of a feedback mechanism for them, because it meant that they could see what we had spiked it with, and then they could tell for themselves whether they found it or not. So it was kind of a bit of a checking to see what they'd done. And so it asked them why they had reported and what was based on their laboratory policies as well. And so I used descriptive statistics for the questionnaires, and then we did like a coding scheme for the reports to see what they did and didn't include. So we got 39 labs involved, which was quite good. And you can see it was from like about 17 different countries. So there was obviously, I was quite impressed. There was like huge representation from France. They were super, super keen to be involved, but the Netherlands were also keen as well. But you can see we got people from all over the world, which was exciting. You can see that most of them were either hospital or university affiliated or both, but we did get a couple of commercial labs involved as well. Most of them were diagnostic laboratories. We thought about excluding the research laboratories, but we decided to include them because we thought it would be interesting and we could see if there were any differences. And about half did in-house and half did outsourced sequencing. And then you can see most of them do their own bioinformatic analysis pathways on site. So just in terms of the unsolicited and secondary findings that they reported, you can see here that most of them did not report the FLCN mutation, and most of them did not report the PAH mutation. And then more than I expected reported the two, or at least one of the MUT-YH mutations. And that was the one where I said they probably shouldn't be reporting that because it's only a VUS. What's more interesting is the reasons why they didn't report them, or the people who did report them. This is the first group. So you can see here that mainly they reported because there could be a treatment or a prevention or surveillance, or because of the age of onset. And so it was because it was a kid and it was a childhood onset condition. That's why they tended to report the FLCN variant. And you can see here that of those who did report from the MUT-YH variants, they predominantly did because it was on the ACMG list. So that was what I was saying before, and because they actively searched for the ACMG list. In terms of why they didn't report the variants, you can see here that with the FLCN variant, their policy was not to report unsolicited findings. So it wasn't because they didn't find it, it was because they don't report it. And again here, this is the PAH, so this was the carrier status. Their policy was not to report carrier status for their laboratory. And then with the MUT-YH, you can see here that they acknowledge that it was a VUS, and they don't report genes, variants in genes that are VUSs if they're not related to the phenotype. So it kind of all made sense, but you can see there was a lot of variation there. So that was quite interesting that we got such varied results, even across 39 different laboratories. And we found that it really depended a lot on what the policies were for that laboratory as to what they did and didn't report. And not surprisingly, the lack of guidance meant that there was variation, but it also seemed that with the ACMG, some people weren't even following the guidance correctly. So that was quite interesting too. So I just want to talk you through another study that I did, which was looking at consent forms, because I think that's an important component about it's another way that the guidelines will influence practice is through what is on the consent forms. So I did a systematic analysis where I searched online, and again, this was quite a while ago, in around 2016, where we searched for consent forms that were either through the online searching or other ones that I had found through some of my other studies with the laboratory scientists. And they had to be actual consent forms, so where a patient was required to sign. They couldn't be like dummy forms or template forms. They had to be specific for NGS, genomic sequencing. They had to be in the clinical context and not research, and they had to be in English so I could read them. And so we used a deductive content analysis for this. So I constructed a predetermined list of things I was interested in coding for, such as things about unsolicited findings, secondary findings, variants of uncertain significance. But then I also had an inductive process where I could add to that list if there was something that was of interest. There was myself coding, but also a couple of other people. So we would discuss any things together if we didn't agree. And we looked at a few different things. So I was quite interested in the terms that people were using to discuss unsolicited findings or secondary findings, what that meant, and what the reporting policies were, etc. So we had 54 forms that met the inclusion criteria, and most of them were for both adults and children, but you can see there that there were some that were specifically for adults or children. Most of the forms we found online were from the US, and also most of them were non-hospital or university affiliated, so predominantly commercial countries. And I highlight both of those because I think that plays a role in some of the other findings I'm going to present. So in terms of the terms that we use to describe unsolicited findings, so out of the 54 forms, 13 of them didn't even refer to unsolicited findings or secondary findings in any way. There was no mention of them. And that's quite a big deal, given it's something that's received so much discussion in the literature. Of those that did, of the 41, 15 of them used the term incidental findings, 9 used secondary findings, and some of them used both incidental and secondary findings. And sometimes that was used interchangeably as well. So it was a bit confusing. And this is why it was good to have someone else co-coding the data to make sure that we were all on the same page. But you can see there that they used a bunch of other terms as well. So you can see it can get very confusing for patients if they're reading these forms. So I said that. So then in terms of what they meant by those terms, when they were referring to when they used the term incidental findings, sometimes they were referring to incidental findings, or what we call unsolicited findings, so where that's identified accidentally. And sometimes they were referring to secondary findings, where it was actually they were actively searching for it. And sometimes it wasn't very clear what they were referring to. And how we identified that was through the kind of description that they gave around what that was. Again, you can see with the secondary findings, sometimes they were referring to secondary findings and sometimes it was unclear. And those that used incidental and secondary findings, it was a real mess. And then in terms of the definitions that they used, so of the 41 forms that did refer to unsolicited or secondary findings, 35 of them provided some kind of a definition or an explanation about what that term meant, which is not bad. 29 of those, it was quite clear, it was much more explicit, so I've got an example of that. So this example of an incidental finding was that in principle, results can occur for all testing techniques that are not directly related to the actual issue, but may still be of medical importance for you and your family, so-called incidental findings. I won't read the rest, but at least they gave a definition that sort of was relatively straightforward. But then 6 provided less explicit definitions. So like this one, the expanded report may contain information on diseases and genes that do not relate to your current condition or may develop many years from now. So don't even refer to the term anywhere near that description. And then in terms of the reporting policies, so of the 41 forms that did refer to unsolicited or secondary findings, there were 15 that said they don't report any unsolicited findings. And then 15 said that they may communicate these sorts of findings to patients. But then sometimes it was really unclear what their policy was as well on these forms. And then in terms of the criteria for reporting, the ones that did actually sort of list why they might report it, it was generally because it was of this medical actionability or clinical significance. In terms of secondary findings, it seemed that 17 out of the 41 actually actively searched for secondary findings. And at the time, remember this was about 2016, that's quite a lot, and that's why I said that I thought this was a bit skewed because of the US basis of the forms, because they're still probably the only ones who are routinely doing this in the world, whereas a lot of places aren't doing it at all. And then just on patient choice, 15 of the forms who offered to report unsolicited findings, of those 7 said that they didn't allow an opt-out. So basically, if you're wanting to have the sequencing done, you are automatically signing up to receive unsolicited findings. 5 allowed patients to choose. So some of them were opt-in, some of them were opt-out, and some of them were both. And then there were 3 of the forms that said it was mandatory for some of the conditions that they would report them back. So this was for things that were childhood onset and clearly clinically actionable, which is sort of in line with some of the Canadian guidelines, which say that you should actually, it's unethical not to return those things because it has clear health benefits for children. In terms of secondary findings, pretty much all of them said that you could opt out, all bar 1 who said that you couldn't opt out, but that was because it was just a cancer panel. So I guess it's a much more targeted test. So conclusions to do with this study were that there was basically a huge lack of uniformity in the practices that we saw on the consent forms, and a lot of variation about whether patients get to opt out of receiving unsolicited findings. So next I want to turn to another study I did, which was looking at what the impact was for the health professionals, who were the people who were basically the coalface of this testing. So I used purposive sampling to recruit genetic health professionals, so that was both clinical geneticists and genetic counsellors, who were actively requesting sequencing for the patients. So they were doing this a lot. They were quite an experienced group of people. So I used semi-structured interviews. I asked them what they received from reports by laboratories, what they returned to patients, and reactions of patients to that information. And so again, this was quite a large study, so I interviewed 31 participants, and that was across 30 interviews. So I had another one where there was 2 people, and English was a bit of a barrier on that one, because they were both French and one was sort of doing a bit of translating for the other person. But so it was mainly clinical geneticists, and again, this study was done around 2017, and at that point, and I think it's changing a little bit now, but mainly clinical geneticists were the people who were ordering these tests, because a lot of genetic counsellors don't have the capacity to actually order them themselves. So that's why there's a bit of a skewing there. But again, I was looking across Europe, Australia and Canada. And I said it was quite an experienced group of people, so they had about nearly 10 years in their current role and about 15 years in the field. And quite a few of them were actually involved in the analysis process themselves. So they had some of them had a bit of a dual role of analysis and also clinical practice. And a lot of them assisted with the multidisciplinary team meetings as well. So that could mean that this is a more informed but also a slightly less representative sample of what's happening in the general public, the general population. So they talked quite a lot about the need to prepare patients for results, and that was for a range of different results. So preparing them for negative results, so no cause being found, preparing them for positive results where a cause is identified, but also preparing them for the fact that variants of uncertain significance are quite common, and talking to them about the potential for unsolicited findings. And there were quite a few concerns about patients not being really prepared enough to receive unsolicited findings. So as this participant said, so despite my best counselling skills and both myself and other clinicians talking about it, they were still very much on the page that we're looking for the cause of her epilepsy, and that's all that's going to look at. I just don't think they were, but I think that as much as we try to prepare our families and our patients for the possible outcomes, I don't think that they really can appreciate that until they're living that experience. So again, I think they were really talking about the need for follow-up counselling and in the results sessions being super important. Some of the participants talked about the patient's experiences of receiving unsolicited findings or secondary findings, and so some said that they felt that some patients found it unhelpful to receive them, but generally the sentiment was that there could be some distress at receiving these results, but that mostly the patients coped really well with the information. So as this person said, how have the families been? Worried but often appreciative of the information, which could make a difference for other family members. I don't want to paint a picture of it being all kind of a rosy situation. I mean, I think some of the families are quite worried. Have I had anyone who's gotten angry about it or become extremely distressed? No. Has there been some distress, potentially about their own health or the health of their other children? Yes, but the families still seem to be not happy that they pursued it, but content with their decision for that information, and some have been really appreciative. And so again, this idea that genetic counselling is important, but it's potentially not really enough to prepare patients, and so you need this sort of good post-test counselling. So as this person said, when I first started doing this, I was like, yep, it's all about the pre-test counselling. It's all about preparing people for possible outcomes and sheltering them and making sure that they're ready for anything and so on. But I think because they're living the experience of some really bad conditions, I don't think they have capacity to think about anything else. Sure, we have to mention it and obviously make people as prepared as possible. I think it's okay to then come back and kind of be there to pick up the pieces in a post-test counselling session. So just to summarise and give you some take-home messages from all of that information I just gave you, which is a lot, so I'm sorry. But I mean, genomic sequencing is really very much embedded in clinical care right now, and it's becoming more and more common in other disciplines that aren't genetics. And so I think we really need to be aware of the impact of mainstreaming. And I think what I've shown is that there's quite a lack of consistency on reporting and also about patient choices. And if that's the case in the genetics community, then I hate to think what that's going to look like in the non-genetics community. So I think we really need to make sure that we're training people who are going to be starting to use these tests to be prepared to do things like obtain consent from participants and also feedback results in an ethically appropriate way. Obviously, laboratory practices and policies vary, and I think that's quite problematic. I think we need to be trying to get more standardisation about what is reported back to patients, because it means that if some patients are getting unsolicited findings and some aren't, and some are receiving variants of uncertain significance and some aren't, and all those things impact on patient care, then it's not really equitable. And the jury is still out on searching for secondary findings. I think that's shifting slightly, but the fact that the ESHG just released this policy last year means that they're clearly not on board with it yet. So I think we need more and more research to be done to try and develop the evidence base to see what these variants do in a non-selected population. And clearly there are some challenges in preparing patients for unsolicited findings, and I think that needs to be part of the training as well. But there's been a whole discussion in the genetic counselling world about should we be like top-ending or back-ending the genetic counselling, and I think I think we can't let go of one or the other, but I think it's a bit situational. So I think with screening sort of stuff, we need to be we need to be automatising a lot of the pre-test counselling and focusing on the back end, because it's obviously like a much smaller proportion of people that are going to be receiving results. But still in the clinical practice, I think we need to be doing both. So I feel like I've talked a lot, so I'm going to leave it there and just acknowledge that this work was done with a whole bunch of collaborators, and I'm very grateful for all the participants that let me speak to them and use their data. So thanks for listening.